Sound test. Test.
Wait, I'm
Sound test.
By the organizer. No.
But I guess we'll wait like till 3:05 if he's not here to
Yeah, it's working. Hey, how's it going?
Good, how are you?
Yeah, flew in for the defense.
I guess it was.
Dave was very happy to see you.
Yeah, he's amazing. Yeah.
How are things going?
It's good. I just submitted my first paper. Got the reviews and just submitted the revisions today. So hopefully it's done.
Great, so nothing, not too bad.
Nothing really bad. It's 2 months of a little bit of,
What's your?
dev files. Yeah, the editor, Claude Spawn was talking about National Academy of Sciences. He's an eye person, so he's familiar with our work.
Is that how he does laughing? So if someone's cleaning your desk yet?
A lot of people leave, so there's a lot of stuff there.
Are you out today?
I'll be
So you just stand to the right of the screen, by the water bottle?
Exactly. Yeah. Right.
Okay, well, let's get started. So I want to welcome hello. Welcome everyone here. This is one of those happy, sad days. So I want to especially acknowledge her committee for coming here and being on our committee for 4 years? 4 years, about. Her colleagues, other students, and most of all her family and friends, so welcome everyone. So the way this will work, just so you know, is we'll let Anastasia present her work, then anyone here will have a chance to ask her questions. Then we'll ask everyone except the committee to leave while we ask her some more questions. You could all go across the street at that point, is that right?
Yeah.
To the courtyard, and we can show you where to go. So I just want to say a little bit about Anastasia. It's been just spectacular having her in the lab. She is one of the people who's made the lab much better as a scientist, as a colleague. She just has a very incisive mind. She's careful with her work. She's creative. She's very thoughtful. She brings a lot of value to it. So a lot of what she's going to show you were things she thought of from reading the literature, thinking about her initial results. And that's what we want from students who can, not just do, but think and create. And she's done that. In part, she has Tom Graber to thank where she started in his lab. And I think she got very good training there in analytical techniques, and then came to the lab, I think, in a very good supportive environment that got her going with what she was doing. She actually started off by picking, I don't know if you're going to talk about the part, BCL2. Okay, so she picked up a project from a leaving postdoc and did some work on that before she really led into her own way. And so this is quite a year for her husband, Michael, for her mother. She got married several months ago and now is getting her PhD, so it's quite a year. And it's really remarkable. Anastasia came to the US at 14 years of age, speaking no English, and here she is getting a PhD from UCLA, so if that isn't the American dream.
I actually did speak a little bit of English, so it's not as impressive. So today I would like to share with you my work on improving nuclease-mediated gene editing outcomes in human hematopoietic stem cells. And here's outline of the talk. First, I'll provide some background on hematopoietic stem cells, on gene therapy and gene editing using CRISPR-Cas9 system, and then I will address some barriers that impede clinical translation of this platform. And then I'll provide some potential solutions that we have worked on over the years, and then finish with conclusions and future directions for this field of research. So hematopoietic stem cells are blood-forming cells that are found in the bone marrow, and we often abbreviate them as HSCs. And they have a unique property to home to the bone marrow and self-renew, as well as differentiate into progenitors, which are collectively known as hematopoietic stem and progenitor cells, or HSPCs, which then give rise to mature blood cells as red blood cells, and then all of the immune system. And this is just another view of the hematopoiesis tree where HSCs are now at the top. And because they have this property to self-renew and differentiate into all blood cell types, if there are certain mutations in any of these cells that cause diseases, correcting or replacing just the HSC population can be propagated down and result in correction of the other blood cells. But it's important to note that this HSC population up here is very rare. They make up only about 1% of all HSPCs, and these progenitors make up about 99%. So making the analysis of these cells very challenging. And here I just wanted to illustrate the power of editing HSCs. So here again are the different blood types, and below each type are the different mutations, the different genes, and mutations in these genes can cause diseases. And by correcting just the HSC population, we can actually correct a number of different diseases. But today I will just focus on disease that's caused by mutation in beta globin gene, or HBB. So HBB is found in human chromosome 11, and this is the 6th codon of the gene, and mutations from A to T lead to amino acid substitution from glutamic acid to valine, which results in the formation of a mutant protein. And this mutant protein, because of the hydrophobic nature of this valine, instead of having normal hemoglobin tetramers, you get the formation of this clumped hemoglobin polymer under low oxygen conditions. And so instead of round red blood cells, these cells become deformed and take on a sickle shape. That's where the disease gets its name. And these cells become rigid and they can clog the capillaries, resulting in severe anemia and other painful crises. So one of the ways to correct this, to treat this disease is by allogeneic hematopoietic stem cell transplant, or kind of the basic bone marrow transplant, where a healthy donor is used and their bone marrow or mobilized peripheral blood are collected, and then these hematopoietic stem and progenitor cells are isolated. And then the patient undergoes conditioning to clear their bone marrow niche of the disease cells, and then these healthy donor cells are infused into the patient, they home to the bone marrow, and they provide a healthy supply of blood cells. Although this therapy is effective, it's not without challenges, and the problems include poor donor availability and a high chance of graft rejection and graft versus host disease. So a way to overcome this is by using autologous hematopoietic stem cell transplant combined with gene therapy, where a patient can serve as their own donor. And bone marrow or mobilized peripheral blood are collected from the patient. Then their hematopoietic stem and progenitor cells are isolated, and you can see here that they contain the disease-causing mutation. And then we can modify these cells in the lab to correct the mutation, and then reinfuse these corrected cells back into the patient after they undergo conditioning. And for sickle cell disease, it's not actually required to achieve 100% correction. Based on studies of allogeneic stem cell transplants, it's been shown that donor chimerism of 10 to 30% has been effective for these patients. So we estimate that gene correction levels of approximately the same range could be curative for these sickle cell patients. So now how do we actually perform this correction? We use something CRISPR-Cas9 system, and CRISPR stands for clustered, regularly interspaced, short palindromic repeats, and Cas stands for CRISPR-associated nuclease. So it's actually derived from a bacterial system, and my work didn't focus on bacteria, but I thought it would just be interesting to show where this technology comes from. So when bacterial cells get invaded by a virus, the bacteria can capture snippets of the viral DNA and store it in these spacers. And then these spacers can associate with a Cas nucleus complex and be guided to the right region in the genome and cut this DNA up. And then they present this on their surface, and that allows them to remember the viruses that they've already been attacked by. So the way this can be repurposed to be used in eukaryotic cells is that you can actually design this short guide RNA that would be complementary to the DNA that you want to target. And you couple it with a Cas9 nuclease enzyme, and it gets targeted to the right region in the genome. And then if Cas9 recognizes this additional PAM sequence, then Cas9 can cut both strands of the DNA. And after the double-stranded break has been made, you can provide the cells with a donor template that contains a corrective base pair here, and using cell-intrinsic pathways such as homology-directed repair, or HDR, the cells can repair that mutation and now incorporate this corrective nucleotide. But unfortunately, there's also a competing pathway, called non-homologous end joining, or NHEJ, which can result in insertions and deletions, also known as indels. And these insertions and deletions are problematic because they result in gene disruption. So when developing this approach, we not only want to improve the levels of HDR, but also want to make sure that we decrease the levels of NHEJ. So the metric that we use throughout the project is this HDR to NHEJ ratio, and we're trying to increase this ratio with different methods. So we start, to model this correction of this mutation, we start with healthy human cells, and here's the site of the mutation, and we deliver CRISPR-Cas9 to induce the double-stranded break as shown. And the way we can deliver this Cas9 and guide is either by plasmid electroporation, in vitro transcribed Cas9 mRNA with in vitro transcribed guide RNA, or a Cas9 protein with in vitro transcribed guide RNA that together is known as ribonucleoprotein complex, or RNP. And we can deliver the donor templates to introduce the desired changes. So we deliver the sickle change. We also change the PAM so that Cas9 cannot recut if HDR has occurred. And then we have two more changes that we introduce that are silent changes that we just use for tracking purposes. And these donors can be introduced as either adeno-associated virus serotype 6 or as a single-stranded oligodeoxynucleotide, or SSODN. So this is just an overview of their experimental design. We start with frozen peripheral blood stem cells, and then we pre-stimulate them for 2 days, and then we electroporate Cas9 and guide RNA, also add the donor either by electroporation or by transducing. And then 24 hours later, we change the media on the cells, put them in expansion medium, and then harvest them 4 days later to quantify the levels of HDR and NHEJ by high-throughput sequencing. But as I mentioned earlier, the majority of the cells are all progenitors, or the majority of the cells are progenitors. So what our readout here is representative more of the progenitors and not of the HSC population. So to be able to evaluate gene editing outcomes in the long-term HSCs, we can use a mouse model where we transplant these cells into an immunodeficient mouse that support engraftment of human cells. These hematopoietic stem cells will home to the mouse bone marrow, and then after 16 weeks, we'll harvest the bone marrow and quantify the levels of HDR and NHEJ. So the levels of HDR that we measure from the bone marrow would be representative of what happened in the HSCs initially. And the way we quantify these edits is if we have like we did in the beginning, we call that unmodified. If we get insertions or deletions, we call that non-homologous end joining, or NHEJ. And if we get either the sickle change or all 4 changes that are present in the donor, we call that HDR. So there is a barrier that impedes clinical translation of this technology, and that is that even though we get relatively high levels of HDR before transplant, so these 3 points represent 3 independent transplant experiments that have been pulled from different studies, where we're getting levels of editing between 20 and 60%, depending on how much donor we use. However, if we look at after transplant, each point here is a different mouse, you see that all the HDR edits go down to below 10%, while NHEJ remains high. And the hypothesis for that is that gene editing, we hypothesize that gene editing happens mostly in the HSC population, is not as efficient as the one happening in the progenitors. So to look at this closer, we sorted the cells into HSCs, MPPs, and progenitors. And the way we did this is based on their immunophenotypic markers, so 34 positive, 38 positive marked progenitors, 34 positive, 38 low are subdivided into HSCs and MPPs. And then from each one of these populations, we quantify the levels of HDR and NHEJ. So if we look at the cells that only got the nuclease, so they could only perform NHEJ, the levels of NHEJ are higher in the progenitors, but it's not statistically significant compared to the HSCs. However, if we look at the cells that did get the donor template and could perform HDR, HDR in the progenitors is significantly higher compared to that in HSCs, and this kind of mirrors the data that we saw in the mouse experiment, while the levels of NHEJ are pretty comparable between different populations. So this poses a major challenge for us in gene editing, because the HSC population is the one that provides the majority of engraftment, and the progenitors don't really have any engraftment potential. So for transplant purposes, we're interested in capturing those cells. However, for editing, the majority of HDR occurs in the progenitor population, while NHEJ is generally similar across different cell types. And we also saw that this correlates with different phases of cell cycle that the cells are in. So if we want to get high levels of HDR, these are the cells that we want to be targeting, the ones that are in SG2M phase of cell cycle. But because this is biology and not quantum physics, we cannot be in 2 states at the same time. So we have looked at different ways to overcome this problem. The first project that I'll talk about is on temporal control of DNA repair. The second project is on improving cell survival. And then the third project is on manipulating DNA repair pathways. So as I showed, the levels of NHEJ are pretty comparable across. NHEJ can happen in any phases of the cell cycle, but HDR is restricted to S and G2 phases. So to verify that this is also happening in hematopoietic stem and progenitor cells, we stain them with a DNA dye, and then we live sorted them into either G1 or SG2M population, and then we electroporated each one of these populations with Cas9 guide and the donor template, and then we measured gene editing levels. So this is just a cell cycle sorting overview, where these are unsorted cells in gray, the orange ones are G1 sorted, and the green ones are SG2M sorted. And when we looked at the levels of gene editing, you could see that in the SG2M sorted population, the HDR was significantly higher compared to the unsorted and G1 sorted cells, while NHEJ was significantly lower. Therefore, there was a significantly higher HDR to NHEJ ratio in these cells. So kind of the 2 logical approaches to improve HDR to NHEJ ratio based on these data is to either decrease NHEJ to decrease nuclease activity in G1 when HDR cannot occur, or to increase HDR to increase the proportion of cells in S and G2 phases. So wild-type Cas9 can make double-stranded breaks in any phase of cell cycle. But the double-stranded breaks in G1 and M phase are undesirable because the only pathway that's available is the non-homologous end joining. So we wanted to coordinate the expression of Cas9 with the phases of cell cycle when HDR can occur. And we did this by tethering this geminin domain to the end of Cas9. And geminin is a naturally occurring protein that targets proteins for ubiquitination and degradation in G1 phase of cell cycle. So by putting geminin at the end of Cas9, we're able to get degradation of Cas9 protein in G1 and M phases while still retain Cas9 activity in S and G2 phases. So to make sure this geminid Cas9 was working as intended, we again sorted the cells and then electroporated wild-type Cas9 or geminid Cas9 into these cells and then measured nucleus activity. And here you could see that in unsorted and G1 sorted cells, the geminid Cas9 was significantly lower than wild-type Cas9. However, in SG2M phase, there was no statistically significant difference between the two. And then we also looked at the different insertions and deletions that are being formed by these two nucleases. So this is a part of DNA that's targeted, and these square brackets represent the guide binding site, and this vertical blue line represents the cut site. And here are the top 15 indels. And on the right, the minus represents deletion, and the plus represents insertion. And here are the percentages of these indels in the two different nucleases, and there was no statistically significant difference between the two. So then we wanted to add the donor template and see how geminid Cas9 would perform in the presence of a donor template and how it performed HDR. So as expected, there were no significant differences in the HDR levels. However, NHEJ was significantly lower, therefore significantly improving this HDR to NHEJ ratio. So now the second strategy to increasing HDR to NHEJ ratio is by increasing the proportion of cells in S and G2 phases. So we used a CDK1 inhibitor called ARO, which temporarily arrests cells in S and G2 phases, therefore allowing us to accumulate more cells in those phases. Here's just a flow cytometry plot showing that we did get more cells with ARO compared to DMSO. And this is just a quantification of that, saying that we do get an increase in SG2M cells and a decrease in G1 cells. And then we wanted to put the two together. So we tested both unsynchronized cells and synchronized with ARO with wild-type Cas9 and geminid Cas9 in each case. And as you can see, ARO led to a slight increase in HDR levels, and geminid Cas9 led to a decrease in NHEJ, and even further decrease in NHEJ with a combination of the two. So if you look at the ratio of HDR to NHEJ, you could see that it was a stepwise increase, and it was significantly better with the double treatment. So as I mentioned earlier, this is all representative of the progenitor cells, but we wanted to look at what happens to the HSC. So we transplanted these cells into mice, harvested them 16 weeks later, and quantified the levels of HDR and NHEJ in the engrafted cells. And what you can see here is that even though the levels of HDR and HDR are lower in after the mouse transplant compared to what they were before, the same trend still holds, that with ARO we're getting more HDR, and with geminid Cas9 we're getting less NHEJ. And therefore we're getting this about threefold improvement in HDR to NHEJ ratio. So to summarize this part, we saw that there's a temporal control of DNA repair. The wild-type Cas9 is active in any phase of cell cycle, but geminid Cas9 is degraded in G1 and M phases, and it's still active in S and G2. We used ARO to increase the proportion of cells in S and G2 phases, and the combination of the two treatments together resulted in an increase in the levels of HDR to NHEJ. So now moving on to the second story in improving cell survival.
Can I just ask a question about that? So, I may have missed this or mistaken this, but I think most of the data, if not all the data that you've shown, is grouping G0 and G1 together.
Yeah.
You're calling it G1, but it's actually got G0 in there as well.
Yeah, I think we're sorting cells after 2 days of pre-stem, so I honestly don't think there's any real G0 left at that point.
I didn't realize that, yeah.
Yeah.
Because that's what my next question was going to be, is because a lot of the cells, especially in the HSC, are actually in G0, aren't they? And
Yeah.
just moving them into G1, so but you don't think that that's sufficient?
I think after 2 days of pre-stem, they're probably not true G0 cells at that point.
Okay.
So we and others in the field noticed that gene editing reagents cause apoptosis and cell death. So to quantify this, we looked at the cells by flow cytometry. So here, a DAPI stains dead cells and an Nextin-5 stains apoptotic cells. So it's single positive for DAPI are necrotic, single positive for an Nextin-5 are apoptotic, and double positive are dead, and obviously double negative are viable. So this is mock, and when we use, so here we use a different nuclease ZFN, but it doesn't really matter for the purposes of this talk. We saw that you get a huge increase in the number of apoptotic and dead cells, and the number of viable cells decreases to only about 23%. So to see what is causing this apoptosis, we turn to the apoptosis pathway. And DNA damage causes upregulation of ATM, which phosphorylates P53 and causes the release of these pro-apoptotic genes into the mitochondria, which results in the loss of membrane potential and the release of cytochrome C into the cytosol. And then the cytochrome C can trigger a caspase cascade, which eventually results in DNA fragmentation and apoptosis. So there have been caspase inhibitors that were used by some people, but it seems like they are active once they're present, but once you remove them, this all built up caspase can still activate apoptosis. So we turn to a BCL2, which targets a little bit more upstream, and it's supposed to block the release of cytochrome C. And this work, as Don mentioned, was initiated by a former postdoc in the lab, Carmen. So what we saw is that if we added BCL2 during our electroporation, we could get a significant decrease in the apoptosis, we could get a lot less dead cells, and we could greatly improve cell viability. And this is just the quantification of the same data. So here are the mock cells. Here are the ones that were edited without BCL2, and you could see that there's a lot of cell death here. And then once BCL2 was present, you could decrease cell death and improve viability. And then we wanted to see if this was actually also improving viability of the long-term HSCs. So we took cells and split them into 3 equal fractions, and then we electroporated the first fraction with ZFN and SSODN, and the second and third fraction with ZFN, SSODN, and also BCL2. And then this represents, so here the gray circles are supposed to represent the cells that have died. So here we only got 4 million surviving cells, and we transplanted them into 4 mice at 1 million cells per mouse. Here we got more surviving cells, but again, we only took a fraction of them. We only took 4 million cells and put them at 1 million cells per mouse. So this represents equivalent transplanted dose. And for this last group, we wanted to have an equivalent starting number, so we transplanted the entire population, so it was about 8 million cells at 2 million cells per mouse. And then after 14 to 18 weeks, these mice were analyzed. So these data represent 3 independent transplants, and you could see that, so these are the levels of human engraftment in the mice, and the engraftment for this arm was kind of different between the 3 different transplants. But overall, you could see that on average, the mice that got ZFN, SSODN, and BCL2 at 1 million cells was lower than that of the ZFN and SSODN, suggesting that the protection that they got from BCL2 was probably affecting the progenitors and not the true HSCs. And here with the cells that got more, the mice that got more cells did have higher engraftment levels. And when we looked at the levels of editing, you could kind of see a trend with higher HDR with the mice that got BCL2, but the NHEJ was also higher. So there was no statistically significant difference in the ratio of HDR to NHEJ. So BCL2 decreased apoptosis and improved viability after gene editing in vitro. However, it did not result in statistically significant improvement in engraftment or gene editing in vivo, suggesting that it's probably affecting progenitors and not the long-term HSCs. So now moving on to the third part, I showed you in the beginning that there are 2 competing pathways, and that HDR uses a donor template to perform the correction. But the decision-making process is not as simple as just whether the donor is present or not. It actually requires a lot of the steps along the way. So here I'll just show some of the important players. If Ku70, Ku80 bind to the site of the double-stranded break, this triggers a cascade of events which eventually lead in NHEJ. However, if mRNA complex binds to the double-stranded break, then you get another cascade of events which either gets repaired, the break gets either repaired by homologous recombination in the case of double-stranded donor, or single-stranded template repair pathway in the case of a single-stranded donor. And there's also a negative regulator of HDR called 53BP1. So a lot of things have been tried by different people in the literature. People have tried inhibiting different steps of NHEJ, people have tried manipulating factors that improve HDR, and a lot of work has been done in the field. But what we wanted to test is this specific combination. So it's been shown that the reason why HDR cannot occur in G1 phase of cell cycle is because it's lacking the formation of this important BRCA1 PALP2 BRCA2 complex. And it's been shown that if you mutate these residues in PALP2, that allows the formation of this complex irrespective of cell cycle. And also providing a constitutively active form of CTIP and inhibiting 53BP1, which is a negative regulator of HDR, can be helpful at initiating HDR in G1 phase of cell cycle. So we thought to try these to try to improve HDR. Another combination that we tried is either ectopic overexpression of RAD52, which is supposed to improve SSTR, together with another inhibitor of 53BP1 to get more single-stranded template repair.
Anastasia, can you remind us, both for the BCL2 and for these manipulations, are you putting those extra genes in constitutively long-term or short-term?
Yeah, so actually, this is going to come up on the slide. So these are the different factors that we tested, and these are the different delivery methods. So we tested lentiviral vectors, integrates effective lentiviral vectors that were pre-transduced 24 hours before electroporation, and then we either tried plasmid or in vitro transcribed mRNA that was co-electroporated with gene editing reagents. And then in the case of BCL2, we just used in vitro transcribed mRNA. So then when we looked at the levels of gene editing, here on the left in gray is the control where no lentiviral vectors, IDLVs or plasmids were added, and you could see that lentis and IDLVs did not result in any improvement in HDR levels. However, plasmid increased HDR, but it also increased the levels of NHEJ, and so the levels of HDR to NHEJ were only slightly higher in these conditions. And the same was true for the other combination for RAD52D and 53BP1, that we also, every time we added the plasmid, we could get an increase in HDR and an increase in NHEJ. So we were a little puzzled by these findings, and we wanted to perform just a plasmid titration experiment to verify that this was true. And we saw we could get an increase in HDR levels and an increase in NHEJ levels as we added more GFP plasmid. And this was, I failed to mention, but this was performed in a leukemia cell line, K562 cells. And then when we moved into HSPCs, we did not observe the same trend. The levels of HDR did not increase with the addition of plasmid. So then we wanted to test the effects of these DNA repair factors now as lentiviral vectors in HSPCs. And we tested them with different donors, SSODN and AAV. But again, with any conditions that we tested, we could not get any significant improvement in HDR or any decrease in NHEJ with the addition of any of these factors. So now there's a lot of different combinations that we tested. And instead of showing you all the graphs for every possible combination, I think it would just be better to summarize it all in a table. So these repair factors were delivered, or repair factors or GFP control were delivered either as plasmids, lentis, or IDLVs in the case of K562 cells, or as mRNA, or these three in the case of HSPCs. And then Cas9 was delivered either as plasmid or RNP, or mRNA, RNP, or plasmid in the case of HSPCs. And then the donor template was either SSODN or AAV. And then for each one of those combinations, we measured the levels of HDR and NHEJ. So in the case of plasmid-plasmid combination, we saw an increase in both HDR and NHEJ. But in all the other combinations that we tested, we could not get any significant improvement in either HDR or NHEJ. and Ellie is currently performing experiments on plasmid-plasmid combination in HSPCs, although plasmids are pretty toxic to HSPC, so even if this turns out to be true, it's probably not going to be a therapeutic approach. So now I'd like to conclude the whole talk. So what I showed you in the beginning is that HDR levels are lower in HSCs compared to progenitors. In the first part, I talked about temporal control of DNA repair in HSPCs, and that gemin and Cas9 NRO can increase HDR to NHAJ ratio about 4-fold. BCL2 decreased cell death and apoptosis caused by the delivery of gene editing reagents in vitro. However, there were no significant effects on engraftment or gene editing levels in vivo. And in the third part, I talked about overexpression of different DNA repair factors, which did not result in any statistically significant improvement in gene editing outcomes. However, we made this interesting observation that plasmid increases both HDR and NHAJ levels when co-electroporated with Cas9 plasmid in K562 cells. So now for future directions, I want to stress that this is kind of a big and important biological problem, and it's very interesting to be studying these cells. And it's not entirely possible whether it's not entirely clear whether it's possible to get high enough levels of HDR happening in HSCs, again, because HSCs are in more quiescent, and as you differentiate them and get them to a state where they're able to do HDR, you lose their engraftment potential. So it's kind of hard to find that balance. But some of the things that the lab is interested to continue testing is looking at additional cell cycle inhibitors, also evaluating alternative approaches to inhibit apoptosis and cell death, and also maybe tethering DNA repair factors to Cas9 instead of providing them ectopically, which would allow for them to be delivered to the site of double-stranded break. And so with that, I would like to thank everyone who has been involved in this project. There's a lot of people to thank for basically the entire Cone Lab, the current people, and all the alumni that I have been fortunate to work with over the years. I want to say a huge thanks to my mentor Dawn, who allowed me to perform research in the lab, and I've really learned a lot over these 4.5 years. I actually came across a notebook that was from my first lab meeting in the Cone Lab, and it was Thule's lab meeting presentation, and it was basically a list of 10 different things and a huge question mark next to each one of them because I didn't understand anything.
Do you know?
I figured it out now, yeah. It was kind of nice to go back and know that I know those things. I want to say huge thanks to my almost co-mentor Roger, because he's been extremely helpful throughout these years, and he has provided a lot of support and a lot of guidance, and I was able to just go to him and bug him for any questions that I had, especially early on. I want to say a huge thanks to my doctoral committee, Dawn, Tom Graber, Gay Crooks, and April Pyle. They have provided a lot of support, and it's been interesting having committee meetings, and you have given me a lot of interesting suggestions during those meetings. I want to say a huge thanks to the Graber Lab, and especially Nick and Buren, who used to be my mentors when I was an undergrad. And I think if it wasn't for Tom's lab, I wouldn't be in grad school right now, because they've really inspired me, and they've taught me so much, and they kind of led me to be here. And then Osbrom was a former graduate student in the lab who remains to be a good friend over the years. I want to say a huge thanks to Carmen, who had initiated the BCL2 project and also kind of been a long friend over the years. And my 3 undergraduates and technicians over the years, Stephen, Danielle, and Ellie, who are all moving forward, and it's nice to see that my mentorship did not discourage them to go forward, and I think I've learned a lot from them as well. Sana, Thule, Bea, and Caroline. So Bea is here. You can see her arm right here. She just doesn't like to appear in pictures, but they have provided a lot of entertainment for me over the years. And my three fellow graduate students, David, Kate, and Richard, it's been fun working with you guys. We're all very different, but I think we learned a lot from each other, and it's been a great experience. I don't have pictures for all of these people, but they're not any less important. The BSCRC floor core, Felicia, Jess, and Jeff have been a huge help with a lot of sorting and flow cytometry experiments, especially Felicia and Jess early on when I was troubleshooting the BCL2 project, and we spent a lot of hours just sitting there and looking at flow data, comparing Carmen's data and my data. So it's been great working with you. The sequencing core run by Jinmin Lee, they have been very helpful. Bioinformatic and statistical analysis performed by Sorel, Michael, and Xiaoyan. A huge thanks to Emily Fitch in the pharmacology department and Jennifer Chung in the MIG department for helping me with all the administrative challenges, and also Jennifer for helping me work with the F31 grant. And then the funding sources, the NIH F31 grant, Dr. Ursula Mendel scholarship, and then the CERM and the Doris Duke that were to Dawn. And then I finally want to thank my family, my mom who's been a huge support over the years, and sorry, who brought me into this country and has loved me throughout this whole journey. My cousin and my uncle, who, although they live in Russia, they are always very helpful. My newly acquired family, my parents, Salam, Marlene, and Steve, who are super nice, and they're very caring people, and they treat me like their own daughter. And this little supporter, who actually knows DNA repair pathways, so he was looking at my slides when I was preparing. And, sorry, I'm very emotional. I want to say a huge thanks to my husband, Michael, who has been with me throughout all these years, on all hikes and trips that we've done together, and has helped me with all the science. He's read all my abstracts and papers and looked at all my posters and has practiced all my presentations, and he'd probably come up here and give the presentation as well as I could. So it's been great having your support over the years, and I definitely wouldn't have gotten here without you. As we conclude for questions, I just wanted to show up this slide. This is a word cloud of my basically 150-page thesis condensed onto one slide. So here, the different size of letters represents how much each word was used throughout the thesis. So you can kind of see some important things as HGR and HGA, gene editing, cell cycle, gene modification, donor template. So those are definitely some of the things that are interesting. And I would like to thank all of you for coming, and some of you have traveled far. So I thank you for being here, and I'll be happy to take any questions.
Beautiful talk. I was just thinking about the timing of the DNA repair factors. Did you ever try them ahead of time, like pre-expression, and then try them? I can't remember.
Yeah, so we've tried them with pre-expressing with lentiviral vectors, and I didn't show here. Ellie has made beautiful Westerns showing that they do actually get expressed. So we know that they are overexpressed. We haven't tried delivering them as mRNA or plasmid ahead of time, just because doing electroporation twice, first for the repair factors and then for gene editing reagents, is toxic to the cells. But we did try lentis, yeah.
Is there a difference in expression just in the resting HSC versus those progenitors of the various factors that you try?
That's a really good question. We haven't looked directly, and I guess you would probably know better from your RNA-seq data.
It up, but I don't
know. I would imagine that they are probably different. Just if you were talking about true HSCs that are truly quiescent, I think they would be different. But At the same time, I don't know if it's necessarily the expression of these factors that is lacking. It could be like post-translational modifications or something that are affecting the formation of proper complexes and some kind of signaling pathways. So it's possible that even though we overexpress these factors, they're still not able to form the proper complexes that they should. Yeah.
Nice talk.
Thank you.
I was wondering, so the stem cells, they make a lot of different cell types, right?
Yes.
And by editing this tiny little spot that's responsible for the sickle cell phenotype, are you expecting to affect any other pathways or any other cell types? Or is it specific to that one?
It's not. So it's not specific to that one, but I don't think it should affect any other cells because you're really restoring it to what it should be in the wild type case. So you're not really modifying it to also if you make any edits that result in, let's say, non-homologous end joining, you disrupt some other genes, that could be a problem. But if you're just correcting the sickle mutation and restoring it back to the wild type, that shouldn't be a problem because the cells already know what the wild type sickle cell locus should look like, or sorry, the wild type beta globin locus should look like.
And then I know you can alter the proportions of the two different changes in DNA, right? But then can you eliminate the other one entirely, or?
I wish we could. Yeah. It hasn't been possible yet. I think that it's not going to be, probably if we come up with some ways to improve the pathways, I don't think it's going to be one thing that, really drastically improves HDR or completely gets rid of NHEJ. I think it's going to be little modifications that altogether will result in some significant improvement that would be therapeutically relevant. But I haven't seen any reports of people completely getting rid of NHEJ yet, but I guess it's maybe possible down the line. Nebula, do you have a question?
No, I think I solved my question.
Okay.
But that was a really beautiful talk.
Thank you.
It was really good. I almost cried.
So I have a couple of questions.
Yeah.
The phenomena you showed with the plasmid increasing DNA
Yeah.
repair in K562, what do you think that's about?
Yeah, so there's two different hypotheses, and I didn't really have time to talk about them in the talk, but it could be that it's either somehow increasing the amount of Cas9 that's made, and what Ellie's actually doing now is co-electroporating Cas9 plasmid with GFP plasmid and looking if you get more protein. It kind of seems like it might be what she needs to do in another experiment to really verify that. Sorry, stealing your thunder. But another hypothesis could be that it's somehow manipulating the two pathways. So it could be that when you induce a double-stranded break, I think the double-stranded break is probably greater than the sum of HDR and NHEJ events because some of the breaks get repaired seamlessly. So it could be that by having plasmid there, you have decreased the amount of those seamless breaks, so therefore the amount of HDR and NHEJ has gone up. I don't really have any evidence to support that, but I don't really have any evidence to support the increase in the plasmid either, but we'll see. I think that's something Ellie wants to continue working on, and it would be interesting to figure out what's doing that.
Is that something you anticipated? In other words, you included that as a control, which is just good science, so that's great. But was it just that you were doing good science and including a control, or you guys thought, oh, adding more DNA could actually have?
No, we actually just use this as a control. The golden rule in the lab that we all know is that every experiment needs a positive and negative control. But we kind of just wanted to make sure it was not doing that, and it was. So I don't really understand how having more plasmid could result in having more Cas9 plasmid formation. I mean, if anything, they'd be competing for Pol2 or anything, and you probably shouldn't get more plasmid or more transcription and translation of the protein.
And you saw it more in K562 than in HSCs.
Yeah, but also in HSCs, we didn't go to such a higher concentration. In K562s, we went up to 10 micrograms. Let's see if I can pull it up easily. So in K562s, we went up to 10 micrograms. But even with 1 microgram, you can kind of start seeing an increase in an HEJ. And I think here one of the replicates was off. But in HSCs, we only went up to 1 microgram because if you started going higher, it ends up being really toxic. But even here, I feel like, you know, if this was real, then we should have seen at least a little bump for the HSCs. So I don't know if HSCs, they probably have additional protective mechanisms which don't allow for this to happen, and maybe with K562s, because they're such a messed up cell line, they can tolerate more genome, I guess, modifications than HSCs can.
And I'm sorry if this was already in your hypotheses, but could the extra DNA be invoking this like a C-gas sting reaction that could be changing?
Yeah, it could be. It definitely could be. The problem is that if it is, I don't know how much it's worth investigating, because if it's only happening, that's kind of why we did this HSC experiment, is because if it's only happening in K562s, it doesn't really help us learn because they're kind of a, you know, messed up cell line, and even if this phenomenon does happen in K562s, it doesn't necessarily help us solve the problem in HSCs. But that was kind of one of the ideas I had, was to look at these different immune responses in the cells and see if we can figure out what it is, and then maybe try to manipulate those in HSCs. Yeah. Yeah.
Do you see any differences? I guess, do you have any evidence to support that if you extract cells from the patient and do manipulations to them, they would perform differently than if you would do them in cell line?
Yeah, we do. I mean, if they would perform differently than they did in cell line. I think cell lines perform generally better, than depending on which cell line you use. I think they're just more permissive of a lot of changes. And we definitely can make more manipulations to cell lines than we can to primary cells. But we could still get pretty good levels, as I showed in the intro, we could still get up to 60% HDR in vitro and culture, which is really good, but it just doesn't persist in vivo. Did that answer your question? Okay.
So a couple more
Yeah.
questions, sort of bigger philosophies. So one of the, you know, an observation that you showed that the level of HDR in the cells in short-term culture is much higher than what you get in the mice.
Yeah.
So there's sort of two main explanations for that. One is that the early stem cells don't do HDR as well, and so they're grafting proportion to their frequency, but they're edited less.
Yeah.
The other is that they're dying more and you're losing them.
Yeah.
So how would you distinguish to try not, because if those either were the problem, there might be ways to attack those. But
yeah.
So how would you distinguish that?
I mean, one way we've done this is by looking at cell sorting, or sorting them into HSCs, MPPs, and progenitors. And even there, we see lower editing levels in the HSC population. We do see some death as well, cell death as well. So I think it's probably the combination of the two. I don't think it's one or the other. But having the data from the sorted cells and seeing that it's lower in the HSCs compared to the progenitors kind of suggests that they just don't do editing as well. I mean, I guess it's good that they don't, for in evolutionarily speaking, but it makes it challenging for this.
And then I guess the $24,000 question is, you know, why were you unable to improve HDR with all the factors? Is it that you tested 8 and it would have been the 9th one, or is it too, you know, what do you think is the problem that?
Yeah, I've kind of thought a lot about it, especially when I was you know, when I was writing and going back and analyzing all the data and trying to see is there anything that we're missing. And I still don't fully have an answer. I feel like we have enough evidence to suggest that these factors that we're providing actually being expressed. But is just having them overexpressed sufficient to get an improvement? Probably not. So we probably need to be looking at, you know, are the complexes being formed properly? So if we're talking, for example, at this protein complex that needs to be formed by providing these factors, is this protein that the cells are making actually able to make that complex? So that's, I think, something that could be looked at in the future. Yeah.
So I've got a couple of questions.
Yeah.
So you were talking about K562 being a messed up cell line, and you're worried about following pathways that may be illuminated in that cell line. Weren't these pathways also illuminated in a cell line that's equally messed up, the U2OS?
That is a very good point. Yeah, yeah.
A kind of a good indicator that that truly isn't the right way to go. It is
Yeah.
better to sit with primary
Yeah,
cells.
yeah, that's a very good point, that maybe these leads that we're following are just not the right targets, and that maybe we should be doing some kind of screen in HSCs and kind of trying to see what's deficient in those cells, as opposed to trying to start there. Yeah, it's a good point.
I've got another one, which
Yeah.
is more kind of, it's the obvious one, which is HSCs, they repopulate our entire blood system, yet they don't do HDR. Can you think of a good reason why that would be the case?
I mean, you think like in terms of in the body as I mean, I think you want to preserve your stem cell niche, and because they're able to self-renew and differentiate, you don't want to be incorporating changes in your HSCs that then will persist for the life. Like if it just happened in the bone marrow, for example, you don't want those changes persisting and then resulting in all these different blood cell types forming that have those random mutations. So it's probably a good protective mechanism for the cells.
So you think they're most likely just to die then?
I think so, yeah. Yeah, which kind of goes back to Dawn's question about are they dying or they're just not doing HDR, and I think it's a combination of both. And I mean, I don't know, we definitely can get HSCs that have been edited that persist, because there are occasionally some cells in the mice, or some mice that do have higher levels of HDR. So I don't think that having higher HDR in HSCs necessarily kills them per se, but I think it's just the process of editing that is probably causing them to die.
Potentially the number of double-stranded breaks and the frequency that they occur.
Yeah, definitely.
Overwhelming.
Yeah. Yeah.
You mentioned something really interesting. You said that although you're delivering these proteins, you're really not sure if the protein complexes are actually forming. And so I was thinking, couldn't you just deliver all the components of the protein complex along with the guide and the Cas9 as a ribonucleoprotein? And do you think that it will persist?
We haven't thought about the strategy. I know BRCA1 is huge. I think it would become a really big complex, but theoretically, if we weren't talking about these particular factors, but maybe, you know, as Roger pointed out, maybe there's other factors that we can figure out that we need to be manipulating, and if they're smaller and delivering those as complexes to the site of double-stranded break, that could work, yeah. Haven't fully thought about that strategy, so I don't know if there's any downsides to that. Can't think of any right away.
I can. Do you think it would actually form the complex without DNA being there, a double-stranded break?
Well, it depends on which factors we're talking about. It depends on how they form. If it's just the two proteins coming together, they probably could. If it's something that relies on binding to the chromatin, then probably not. Yeah.
So just, I think, kind of leading off those questions as well, sometimes it's easier to break something than to make it work better. So did you guys think of slowing down the other side of the pathway?
We did, yeah. So what we actually tried in the beginning was my very first project in the lab is inhibiting ligase 4. And there were a series of papers that were published on this really good inhibitor, which then a lot of further papers kind of disproved or showed that it didn't work. But I think the problem, and again, they were doing work in cell lines and it worked really well for them. I think the problem is that by the time you get to the step, the cells have already committed to the pathway. So it's kind of targeting it very late. So if the cells are not able to finish, they're probably just going to die. So which is not a good strategy for us. Kind of ideally, it would be to target Q70, Q80, but they're some of the most abundant proteins in the cells. So actually inhibiting them would be very challenging. But it could be that inhibition of several of these factors all together maybe somehow will push cells to perform HDR as opposed to doing an HEJ. Yeah.
What age mice did you use?
Which mice?
What age?
Oh, we transplant them at 6 to 9 weeks old.
And then collect them 4 months later?
Yeah.
So do you know if the stem cell niche changes with age at all?
It probably does, and but we're sublethally irradiating them, and we're providing human cells at home to the bone marrow. But yeah, the niche probably does change. Are you thinking that it could affect that maybe we are getting edits, but they're just not persisting because of the changes in bone marrow niche?
Yeah, I guess, or maybe using younger mice or older mice, or I mean, how does that affect the results?
It's kind of a standard assay in the field. I don't know, Gabe, if you have any thoughts
Well,
about
I'm
it.
just thinking, I think that's a really interesting question, actually, because the busulfan data, busulfan versus radiation data that Ken Dorshkin has been reporting, and that, you know, the so-called aged stem cell that doesn't engraft as well or whatever, it varies depending on the conditioning, and you do wonder if that might affect things at all.
I mean, we are still getting high levels of NHEJ, so if anything, I don't know how the niche would preferentially affect the cells that have done HDR versus NHEJ. So if we were getting no edits at all after transplant, that could be a good reason. But I mean, I'm not saying that's not true, but I'm just kind of thinking it's not likely to affect it that much, just because we're still seeing the other pathway being edited, or cells being edited via the other pathway that are present.
Unless there's something about an HSC that gets HDR.
Right, so I was going to ask, actually, Gabe, this question. I can't remember how it goes, but isn't there a difference in the homing of stem cells that are cycling versus the ones that are not? Ed Swire has done stuff. I can't remember which is better, but so
it's better not to be cycling.
Yeah, it's definitely better to be in
G0 HSCs engraft better than
right.
G1s.
And so the ones that are going to get HDR are not the G0 ones.
Yeah, so
is homing or engraftment part of the problem?
Yeah, so it could be
the assay.
Yeah, it could be going back to this, that by pushing cells to cycle, we're also losing their engraftment. We're giving them an opportunity to do HDR, but they're not able to engraft. I keep mentioning this. Jacob Coren had an interesting bioarchive preprint maybe about a year ago now, where they took HSCs, they pushed them, they allowed them to cycle, did HDR, and then they used some molecule to put them back supposedly into quiescence. And at that point they didn't have any mouse data, but I've been kind of waiting for a paper to come out, and haven't seen anything yet, but maybe that's going to work. I'm not really sure whether it's possible to put cells back into quiescence, because they were showing cell cycle profiles, but I don't know if being in G0 necessarily means that the cell's quiescent. So, or being in G1 necessarily means that it's engraftable in quiescent cells. So we'll see if that ever comes out, but that could be an interesting strategy to kind of allow them to cycle, to give them an opportunity to do HDR, and then to put them back.
Okay, any other questions? I think you've handled those very well. So then, if your committee stays behind, and the rest of you can go across the street and start the refreshments.
I know you're a unit, but you're not that.
No.